She's a Mona Lisa. Everyone's lining up to see her. She's a Mona Lisa.
Assalamualaikum. Today, I will show you how I made my 3D bacterial cell model. Let's start! First, I cut the styrofoam. I made 4 pieces of 3x5 and 5x9 inches. Since the styrofoam I use is thin, I would have to combine it with white glue. Second, I use a blade that I cover the other side with stick glue for my safety to curve the edges. Third, I cut its parts out of the styrofoam excesses. I made pili out of it, a plasmid, food granules, and ribosome. Then, I used the wire of a disposable face mask for making the chromosome. After making the structure and its part, I covered it with tissue paper with white glue mixed with water using a paintbrush. When it's already dry, I painted it. I placed cellophane under my materials to avoid getting the wood stain. I used jar cups as my palette so I could mix the colors since I only have 2 colors, green and yellow. I painted the capsule pili, flagellum, yellow, and light yellow for the cytoplasm, while I painted the cell wall dark green and light green for the plasma membrane. As I mentioned, I only have green and yellow paint. So for the plasmid, I used my pink lipstick and red lipstick for the food granules, then blue highlighter for the ribosomes. Lastly, I used stick glue for the pili and flagellum to stick it to the capsule. Ta-da! My 3D bacterial cell! Pili in plural term allow bacteria to colonize environmental surfaces or cells and resist flushing. The pilus have a shaft composed of a protein called pilin. The flagellum is a motility organelle that enables movement and chemostasis. The capsule camouflages the bacteria from the immune system by mimicking the host tissues and makes the bacteria resistant to complement invasiveness. It protects cells from desiccation and toxic metabolites in the environment. The cell wall has multiple functions during bacterial growth. It maintains the bacterial cell's integrity and shape as well as restricting internal turgor pressure. The plasma membrane regulates the transport of materials entering and exiting the cell. The cytoplasm or protoplasm of bacterial cells is where the functions for cell growth, metabolism, and replication are carried out. The cytoplasms contain cell structures such as ribosomes, plasmid, a chromosome, and food granules. Plasmid. It can transfer from one species of bacteria to another through a process known as conjugation. Ribosomes. It functions as a workbench for protein synthesis, whereby they receive and translate genetic instructions for the formation of specific proteins. Chromosomes. It performs the complex task of efficiently compacting DNA while supporting gene regulation and proper DNA segregation. Food granules are the parts of the cell that store the cell's energy reserves as well as other significant metabolites.
That's it! Thank you for watching this video. If ever you like this video, then click the like button, subscribe, and hit the notification bell. To more tutorials with Rahza.
She's a Mona Lisa.